I said that Zheng-Yi couldn't be here from Boston, but we're hoping that he's going to Skype in right now. So let's see if that happens.
Well, first of all, I really thank the organizer for the invitation. And this is a meeting I've never been to, and this is a meeting I'm really looking forward to. Unfortunately, my flight was canceled because of weather conditions. So, but through the wonder of technology, I think the way we communicate now, we can still hopefully reach the goal we initially set up to do. So what I want to talk to you about today is really about how we want to take the information we learn from discovery of genetic deference gene to a new direction, that is to use information to develop a treatment for the future. So we all know from previous talks that we use it in a year for hearing and the vestibular organ for the balance. And the process in a year for the hearing is conducted through the sensory cells called the hair cells, as shown here. And in particularly is through the particular structure called the stereocilia, which are located on the top of each hair cells. And due to the signal transduction and channels function, the signal we hear as a sound will be converted to electrical stimuli, and ultimately from in the ear will go into our brain so we can sense the sound. So the previous talks have clearly illustrated that genetic deference is very complex, involving multiple genes and many mutations, and also in terms of manifestation in so-called non-syndromic deafness, where we primarily see the phenotype of hearing loss, where in syndromic deafness we see hearing loss in addition to other phenotypic changes such as heart disease, blindness, and so on. And this is the number I plugged in. Actually, I heard the previous talk, I realized it's already outdated. It's close to 120 genes to be discovered. But this, I have to say, is a kind of moving target, because with every day, every month, every year, the more and more different genes will be discovered. And in addition, from the animal studies and also some genetic calculation, we estimate by the community that there are probably far more genetic deafness genes are waiting to be discovered. So we have the number, it's probably close to 800, if not more. So despite the rapid discovery of hundreds of deafness genes, really the treatment for genetic deafness has been, kind of lag behind. This is not only for the genetic deafness, but also for, in general, genetic diseases. So currently, the treatment for genetic deafness, especially profound deafness, is primarily using cochlear implant, which has a tremendous success. But in the future, we're really envisioning many different types of treatment paradigm for genetic deafness, such as gene therapy, cell replacement therapy, or just small molecule-based drug therapy. So one of the main topics today is really for me to describe how we utilize, or in the field, how everyone use the mouse model or the animal model to study genetic deafness. So why do we use the mouse model in general? Well, traditionally it has been because to study any genetic conditions, and people be using Drosophila, zebrafish. However, the species were very useful. We learned some really great insight from biological mechanism. We still think there's a big gap between from those animals to human, and the mouse model has turned out extremely valuable biological tool. That is, we can do genetic manipulation in mouse model. We can make mutation of genes or mimic a human condition and understand what all the phenotypes could be due to the mutations or various genetic manipulation to the phenotype. And in addition, such a model can be used as a paradigm for developing new treatment. So for example, this is a mouse model. If you look at a mouse, in a year on the right-hand side, it has a 4-row outer hair cell, 1 inner hair cell, and on the left, the human cochlea. You see there's a lot of similarities based on one aspect, that's just the hair cell distribution and configuration. But importantly, with the animal model, such as mouse, when you have a genetic mutation affecting hearing, as shown here, you see the phenotype, the disease phenotype, which we cannot get from human study. Human in a year, we cannot get the samples, and I would say vast majority of genetic deafness, we really don't have a human pathology to examine. But in mouse, it's very clear. For example, on the left-hand side, you have a very normal hair cells. On the right-hand side, where you see the degeneration of the hair cells, this will tell us what's going on, and will also provide information for the development of treatment. So we know the gene mutation can cause hearing loss in mouse, very similar as in humans. So provide a tool to study hearing loss pathology, and due to the many years of work and there's many mouse models, practically for every human genetic deafness is a mouse model, and probably far more mouse genetic deaf model out there than human, which can be potentially useful for the future. The other thing is mouse has a relatively short lifespan and easy to breed. We can generate a large number. That would be extremely valuable for our testing, development of treatment. So, as the previous talk has shown, that there's two major genetic deafness. One's recessive. So what recessive means is you really, for each gene, we carry 2 copies. And if one copy has a mutation, and yet you are not being affected, that means that mutation is recessive and you are just a carrier. Like in this diagram, the mother and father are the carrier. However, by probability, if you pass on to the next generation, quarter of the children, which on the far left, that will have inherit both mutant copy of the gene. In this case, that child will be affected, where 50%, which is 2 children in the middle, they are the carrier, and another quarter, which on the right, will be completely normal. So for this, because we lack of a functional copy of the gene, so for treatment, the current standard approach is to use the virus. This virus is called AAV, adeno-associated virus. Itself doesn't cause any disease, but we can use it to carry the gene, functional copy of gene. And as shown here, then afterwards, we could potentially introduce them into the affected individual. As a result, due to the extra copy of the functional genes, we can hopefully restore the function, including hearing. So in the last few years, there is a large number of studies to show you such gene supplemental approach to treat genetic deafness in mouse model, and they've been extremely successful. And those approaches are well on the way being developed for clinical applications. The other type of genetic deafness is called dominant. That means for each gene, you have 2 copies. One's normal, one's mutation, mutant copy. But due to the malfunction mutant copy, it actually interferes with the normal function of the normal gene. As a result, it will cause genetic deafness. So in this particular case, even you only carry 1 copy mutant gene, you could be affected. And in this diagram, the father is affected and half the children are affected. So for this one, by simply introducing a functional copy of gene will not be sufficient, because what we really need to do is to eliminate the function of the mutant copies. So there's some technology out there for such a process, such as RNA interference. Basically, that is through a different pathway which will interrupt the production of the mutant protein through the process. So in this case, we can use RNA-based approach to disrupt the mutant copy so the normal copy can function properly, and as a way to restore the function. And this kind of study is not easy to do and extremely difficult. So there hasn't been as many studies as recessive genetic deference treatment. So, but the pioneer in the field, like Richard Smith and James Lindsay, they have shown the possibility such thing can be done. So today, another thing I'm very excited to talk about is so-called new technology called the CRISPR technology. So previous genetic treatment paradigm I talked about is really by introducing new material, either functional gene or disrupt the mutant gene. But on the other hand, the genetic mutation on your DNA is still there. They can still create, wrong protein or in a way affect your gene or cell function. But this new technology is called CRISPR. It's class of the regular interspace short palindromic repeat. So what it does is it actually act as an editor on your DNA to change the DNA configuration as a way for treatment. So the process actually is relatively simple. That involves a protein, which is shown red here, and it's called Cas9, and the guide RNA. Guide RNA is a piece of RNA which can recognize the mutation a person has. So in this way, they can function as a gene editing process. And here I show you a very short video to introduce this technology.
CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut and paste tool for DNA editing.
So, as is shown in the video, this technology can really actively editing the DNA in our body. As a result, we could offer a kind of treatment that is permanent. So CRISPR can be used for two ways. One is as shown here on the left-hand side, and one is to repair the gene. If you have a mutation, you could potentially repair. On the right-hand side in this figure, that it shows you can also disrupt the gene. Okay, so that means if you have a mutation, as earlier on I mentioned, such as dominant genetic mutation, so this one, CRISPR technology, is extremely powerful because it can disrupt the mutation as a result to restore function. So CRISPR technology is one really transforming technology out there and being rapidly developed for treating a lot of diseases such as cystic fibrosis, Duchenne muscular dystrophy, hemophilia, Huntington disease. We have been actively working in the field trying to use this technology to develop treatment for genetic deafness. And here I'll just tell you one example we've been working on. That is, there's a human dominant genetic deafness called DFNA36, which is due to a mutation. It's called a Beethoven mutation, but actually has nothing to do with Beethoven. Simply, in the field, we're trying to give some interesting name to this mutant. So it's called a Beethoven mutation, actually happening in the TMC1 genes. This is one of those large Chinese family shows multi-generation suffering from the disease transmitted as dominant. That means they only carry one mutant in their cell. Here it shows on the top the normal copy, on the bottom the mutant copy. So the happening is from T to A, that causes the mutation. So the gene is called TMC1, the function possibly as a transduction channel located in the stereocilia and the hair cell, so it's absolutely essential to hearing. And in the mouse model, Karen Steele and co-worker have shown, in the mouse model, without the gene, the hair cell will die, the animal will exhibit the hearing loss. This mutation in animal model is actually exactly the same as in human. So we tried to develop a CRISPR-based rescue for the study. What we did was, as I said, CRISPR technology, we only need two components, which is Cas9 and the guide RNA. So we combine them and with lipid formulation and inject mouse mutant. And through the DNA sequencing, we can clearly see that you can see the A, that is a red, that's a mutant. Through the CRISPR, we can create the so-called indel, that's the insertion, deletion, to disrupt this particular mutation. And the sequences just show such a data. But importantly, this is animal. In the Beethoven animal, they have profound hearing loss. This is by ABR and acoustic brainstem response. And it shows that you have to give a different noise at a different frequency. And in order for them to hear, you have to give a very loud noise for the animal to hear. But if you treat them with CRISPR complex, we can substantially lower the hearing ABR threshold to a stage they can hear relatively normally. Importantly, if you look at the inner ear, this is from the same mouse. On the left is uninjected ear. The green means all the hair cells. But you can see on the left-hand side, untreated ear, you can see a large number of cells died, disappeared. But on the right-hand side, after treatment, the cells are being preserved. So not only can restore the hearing, but also can promote the cell survival. So what's the next stage for such a work to really move to clinic? So we know in mouse model, create many mouse models which are great for the study, but a mouse model still has a lot of limitations. That is, in the ear is very small, it's very difficult to develop like deliver procedure and to study various parameters which are critical to clinical application. So recently we have turned into study a pig model. So pig has been used for studying many diseases, and we want to introduce a pig for such a study for the inner ear. So the pig inner ear has the same cell structure, inner ear structure, but this is one of the mutant pig you can show on the bottom. When you have a mutation, you can see the hair cell defect, just like in a mouse. And the pig is very big, can be used for a lot of development. So that's why this model will be extremely useful. And the CRISPR technology not only extremely powerful to develop and treatment, but also can be used to create, such as the pig model for genetic deafness. So we have used the CRISPR technology to create a Beethoven, the one I just mentioned to you, as shown here. We create the exactly same mutation as a mouse, as a human, and subsequently we're going to treat this pig with CRISPR technology to restore the hearing. And in addition, we have also established the human cell line from this patient, and the cell line can be used to differentiate into human hair cell, carry the mutation. So we can also study by CRISPR technology whether our treatment is effective in actually human hair cell, but in dish. So in summary, I think I've told you about how CRISPR technology can open up a really new route to treat genetic deafness. And we have shown that the animal model all the way from zebrafish to mouse to pig are extremely valuable to the development for any treatment for genetic deafness. And CRISPR technology can also be used to create an animal model which is unforeseeable and in just a couple of years ago, but now it will become such a reality, which in the end will accelerate the process to develop the treatment. And finally, I'd like to thank my group with Mass Eye Ear, Harvard Medical School, for their work, and also our collaborator from David Liu's lab, Broad Institute. And also echo previous talks that shows the collaboration, especially some international collaboration, is absolutely essential to the success of our effort. So also the peak model and the future development treatment, we really have to work closely with our collaborators. And in the end, we can help everyone throughout the world. And thank you for your attention.